Good morning everyone present here, respected teachers and all my dear friends. My name is Sunil Rao. Today we are gathered here to deliver some information regarding G6PD deficiency. So, the full form of the G6PD is Glucose-6-Phosphate Dehydrogenase, the enzyme which is found in all human cells. It plays a very important role in pentose phosphate pathway and helps the cells from oxidative damage. What is the function of G6PD? The G6PD converts the glucose-6-phosphate into 6-phosphoglucono-delta-lactone and produces NADPH, which maintains the glutathione in its reduced form and protects RBCs from oxidative stress. The detailed version of glucose-6-phosphate dehydrogenase is its deficiency is genetic disorder that affects RBCs and can lead to hemolytic anemia, especially under certain stresses. What is G6PD deficiency? So the G6PD deficiency is caused by the mutations of G6PD gene, which provides instruction for making an enzyme called glucose-6-phosphate dehydrogenase. These enzymes play a very important role in protecting RBCs from the damage caused by the oxidative stress. The HMP shunt pathway. The HMP shunt pathway is an alternative pathway for glycolysis and disease cycle for the oxidation of glucose. It is more anabolic in nature since it is concerned with the biosynthesis of NADPH and pentose sugar. The pathway occurs in liver, adipose tissue, adrenal gland, erythrocytes, lactating mammary gland. The pentose phosphate pathway, the substrate required here is glucose-6-phosphate and the cofactor is NADP plus, which further forms NADPH. The enzyme required here is glucose-6-phosphate dehydrogenase and the product form is 6-phosphoglucono-delta-lactone. What if there is no G6PD present? So the phosphoglucono-delta-lactone will not be formed and majorly the NADPH synthesis will be affected. Being the only source for NADPH, the production being hampered, this will affect the reaction that requires the NADPH as reducing agent and increases the oxidative stress. The functions of NADPH. It is a free radical scavenger which protects the RBC membrane integrity. GSH, which is glutathione of reduced form, detoxifies the hydrogen peroxidase catalase reaction. NADPH is responsible for the regeneration of reduced glutathione from oxidized one. Hydrogen peroxide chemically damage unsaturated lipids, proteins and DNA, prevented to larger extent through antioxidant reaction involving NADPH. The maintenance of concentration of reduced glutathione. It also keeps the ferrous ion in its Fe+2 form, so the accumulation of Fe+3 is prevented. The genetical basis of G6PD deficiency is it is X-linked recessive inheritance, most common in males, as the male has only one X chromosome and the female has two. The female will be the carrier for this G6PD deficiency, and it is more prevalent in African, Mediterranean, Middle Eastern and South Asian descent. The triggers of hemolysis, that is RBC breakdown. People with G6PD deficiency may experience symptoms after exposure to certain medications like sulfur drugs, anti-malarials, foods like fava beans, infections especially bacterial and viral, chemicals like naphthalene. The symptoms of G6PD deficiency is fatigue, pale or yellow skin, dark urine, rapid heart rate, shortness of breath, enlarged spleen in some cases. The diagnosis of G6PD deficiency is G6PD enzyme activity assay, CBC, that is complete blood count, reticulocyte count, peripheral blood smear may show white cells or His bodies. The management of G6PD deficiency is avoid known triggers, treat infection promptly, blood transfusion in severe cases, education of medication safety and dietary restrictions. The G6PD deficiency has affected over 400 million people worldwide, offers some protection against malaria, which explains its prevalence in malaria endemic regions. The importance of G6PD is it is important as it is the only source of NADPH. The NADPH is crucial for the neutralizing reactive oxygen species, supporting fatty acid synthesis, enabling detoxification reaction in the liver. Further, the information will be delivered by my friend Sushi Jadhav.
Hello friends, now let's look towards the clinical correlation. G6PD deficiency is X-linked recessive genetic disorder, affects over 400 million people worldwide, causes hemolytic anemia in response to certain foods, example fava beans, medications like sulfur drugs, antimalarials and infection. For better clinical case visualization, we are presenting the case on favism caused due to G6PD deficiency. Drama based on the following topic. Name, Don't Blame the Beans, a G6PD comedy. Following are the characters. Dr. Sumit, senior physician, calm, kind and walking medical textbook. Dr. Sammy, first year resident, enthusiastic and very confused. Dr. Suraj, overconfident resident who thinks Grey's Anatomy is real life. Nurse Joy, on the top of everything and delivers savage one-liners. Helper Hari, the dramatic helper who thinks he is Sherlock Holmes. Mr. Lalaram, the unfortunate patient. He just wanted healthy snack. Mrs. Lalaram, Mr. Lalaram's wife. She is loud, proud and always 2 seconds away from the fainting. Scene first, emergency begins. Mr. and Mrs. Lalaram are having delicious dinner of fava beans.
भाई वाह, क्या जादू है तुम्हारे हाथों में। चेंगा भी वैसे ही पकाती हो जैसे पकाती हो मुझको बातें में।
जेवना चुपचाप। A day later. At midnight, Mr. Lalaram had difficulty in breathing.
सगू, सगू कुठ गेली सगू? ए सगू, आली का? सगू,
बेटा काय झालं तुला?
श्वास येत नाही, त्रास होतोय मला, बोल लवकर.
हरी बोल काय झालं?
काय झालं दादा?
अरे वाटलं काय? श्वास येत नाही.
चल हॉस्पिटलला गोडे सरांच्या हॉस्पिटलला जाऊ. उठ. झोपला झोप.
Mr. Lalaram lying on hospital bed.
काय झालं तुम्हाला?
Nurse Joy checking his vitals.
डोळे वर करा. येलो आईज. जीभ बाहेर काढा. तेल टाळून. हात दाखवा. येलो नेल पेन. acute tachycardia. Pulse 110. बसा. तुम्हाला तुमच्यात आणखीन काही बदल वाटतोय का?
श्वास घेण्यात खूप त्रास होत आहे आणि रंगी पण काळी दिसत आहे.
हे घेऊन जा.
Enters lab technician Sumetha. Take away sample of the patient.
देखते हैं व्हाइट कोट वाले क्या गुल खिलाते हैं, लिखकर देते हैं टैबलेट या सिरप पिलाते हैं।
देखेंगे ना दादू से क्या करते हैं।
गुड मॉर्निंग डॉक्टर. आपल्याकडे एक पेशंट आला आहे. ऑन बेड नंबर 5, मेल इन मिड 20s, थकावटीसोबत पिवसळ डोळे आणि काळी लवली.
अरे बाबा असं काय खाते होतास? तुझे सिम्पटमचं प्रत्येक चॅप्टरच्या एक पेजवर दिसत आहे. मला पण सांगते.
तर खराब आहे हो तुमची. लालाराम नाव माझा, वय 26. डॉक्टर साहेब, काल रात्री मी वालाच्या शेंगा खाल्ल्या होत्या. त्यानंतर माझी लघवी कॉफी सारखी दिसत आहे हो.
अरे मेल्या, सांगितलं होतं ना तुला नको खाऊ त्या शेंगा. पडलास ना आजारी.
एक मिनिट डॉक्टर साहेब.
डॉक्टर साहेब इकडे आहे.
हा साहेब, एक मिनिट. तुला स्वतः त्या शेंगा ठुसायच्या होत्या म्हणून मला मना करत होतीस. मी एकदा पण बिमार पडेल असं तर निघाला नाही.
चुप बस हलगड वाळाच्या. कुठे पण काय पण बोलतो. लाजला वाटत.
आणि वाटत
Latest lab technicians will update the blood reports of patients. Seen changes to suspicious beings.
So how do we diagnose? Okay, the G6PD deficiency is.
Use mine.
What do you know about the G6PD deficiency?
Use mine.
G6PD डेफिशिएंसी में G6PD डिफिशिएंट होता है.
वाह, इसको तो अभी के अभी आईएएस घोषित कर देते हैं.
थैंक यू सर भाई. बट
Don't mind. It's not तुमच्या रेड ब्लड सेल्सकडे काही टूल्स नाही आहे सर्टिफिकेट्स हँडल करण्यासाठी, जसे की वाराच्या शेंगा झालं, मेडिकेशन्स आणि काही इन्फेक्शन्स.
ह्याच्या पेशी नकळला सर ह्याच्याच सारख्या.
Seen treats clinical skills.
So how do we diagnose it? Good question. So the first, the clinical suspicion. Look out for anemia and other looking characteristics of the blind patient. Second, labs. Investigate the high bilirubin, high elevated LDH, a blood smear showing white cells or Higgs body, and finally the confirmatory test is G6PD enzyme assay.
मला वाटलंच होतं हा सगळा एन्झाईमचा गफला आहे, कारण सगळ्याच नखरेबाज आहेत एन्झाईम.
आणि तू शेक्सपियरची चौथी अवलाद.
Seen 4. Management money.
Management is simple. Avoid triggers, hydrate and monitor. If anemia is severe, transfuse.
तर मग वाल्याच्या शेंगा पण मी तर 5 किलो आणलं होतं.
भरून घे त्या शेंगा तुझ्याच. नदीत फेकते मी त्या.
आपल्यासाठी एक शिफारिश, आजपासून वालाच्या शेंगा बंद.
Exactly. Avoid drugs like sulfa antibiotics, antimalarials like primaprine, and some pain medications. I'll give you a list.
So it's about reducing oxidative stress. यही तो जानना होता है.
In other words, no pills, no stress, and no self-medication. Zip height, discharge, and drama.
जीने की थी तमन्ना मेरी, क्या रह जाएगी वो अधूरी. दूर हो गया जो मैं उससे, सह न पाएगी वो दूरी. सोचा नहीं था मैंने शेंगा कर देगी मुझको बीमार. क्या खाते खाते हो गया मैं एनीमिया का शिकार.
नहीं बड़ा, आज नहीं, अगली बार.
सगळं काही गुगलवर सर्च करणं बंद मारारे. टपकन एखाद्या दिवशी एखाद्याला माहित पण नाही पडणार. एकही मॅनेजमेंट सुट्ट्या पण देत नाही. घरी जायचं मला.
So what the story, moral of the story was?
Know your enzymes, respect your blood cells, and avoid the suspicious beans.
Here is educational takeaway message for you. G6PD deficiency is common in some population, especially males, because its X-linked. Triggers include drugs, infection, and fava beans. Management is mostly supportive and preventive. Don't forget to educate your patient before they educate themselves on YouTube. And always ask, have you had suspicious legumes today? Don't blame the patient, blame the beans. Now Sumedha will tell the summary of.
I'll summarize it for you. G6PD, glucose-6-phosphate dehydrogenase deficiency, is a genetic disorder affecting the enzyme responsible for protecting red blood cells from oxidative stress. When individuals with G6PD deficiency are exposed to certain triggers, example specific medications, foods, or infections, it can lead to oxidative stress and hemolysis. Oxidative stress, damage to red blood cells. Hemolysis, breakdown of red blood cells. Symptoms can vary in severity and may include jaundice, yellowing of skin and eyes, fatigue, feeling weak or tired, pale skin due to anemia, dark urine from bilirubin buildup. Clinical findings. Diagnosis often involves blood tests to detect enzyme deficiency or hemolysis, genetic testing to identify mutations, management and prevention. Avoiding triggers and managing symptoms are key. Firstly, avoid triggers. Second, monitoring. Third, supportive care. Avoid triggers, certain medications, foods, example fava beans, and infections. Monitoring, regular checkups and blood tests. Supportive care, managing anemia and other symptoms. Next will be continued by the topper of the batch and topper of the board, the one and only Sumit Kadu.
Hello, good afternoon everyone. So let us come to an ending and conclusion. So with proper management and avoidance of trigger, many individuals with G6PD deficiency can lead normal lives. However, severe cases may require ongoing medical care. Anyways, this is the end. But let I tell you, as you are all very studious and very exceptional student, here are some questions that you might find came to your mind. Okay. The very first question is, why is the urine dark color in G6PD deficiency? So it is written there, but I will let you answer. Anyone? No? So to put it in a simple language, dark color urine occurs due to hemolysis. That is, G6PD deficient cell can't produce enough NADPH which is needed to protect the red blood cells from oxidative stress. Under stress, red blood cells break down. They release the hemoglobin, is broken down into bilirubin and other pigments. These pigments, especially hemoglobin or urobilin, are excreted in urine, making it appear a dark or tea color. Second question, what are white cells or hand bodies? I'll let you answer once again. Anyone?
For this group will get the marks, the other groups commute the marks. What is the answer?
So here's the answer. White cells and hen bodies are both abnormal red blood cells, features seen in conditions like G6PD deficiency, especially during oxidative stress. Here's the quick explanation of each. Hen's body. What are they? The clumps of denatured damaged hemoglobin inside of red blood cells. Simple. Cause oxidative damage from reactive oxygen species when NADPH is insufficient. Appearance, as seen in dark confusion in red cells, is patient stain. Due to this, the urine appears dark. Okay. Next, the white cells are also known as plasmacytes. What are they? Red blood cells that look like someone has taken a bite out of them. You can imagine that. If someone has taken the bite out of it, it will be looking like an apple, eaten apple. Because the spleen removes Hen's body from the red blood cells by biting them out, leaving this characteristic shape. Appearance seen on regular blood smears, often alongside signs of hemolysis. Here's the next question for you. What is G6PD enzyme assay test? I'll let you answer once again. So, anyways, the G6PD enzyme assay is a blood test used to measure the activity level of enzyme G6PD in the red blood cells. It is primary diagnostic test for detecting G6PD deficiency. What is the purpose of test? To confirm the G6PD deficiency in individual with symptoms like unexplained hemolytic anemia, dark urine, and jaundice. To screen people before prescribing oxidative drugs, example Primaquins, which is an anti-malarial, and sulfatase, which is like antibiotics. How does it work? A blood sample is taken, a lab measures how much NADPH is produced when glucose-6-phosphate is present. Low NADPH production is equals to low G6PD activity, indicating deficiency. Test interpretations are simple. Normal active means no deficiency, and low activity means G6PD deficient. In mild deficiency, resetting may be needed after hemolysis subside because young RBCs may still have normal G6PD level. One last question. How does the G6PD deficiency provide resistance against malaria? As it was told by my friend Sumit, but I'll ask you anyways.
अरे बोला ना एखादा तरी आन्सर करा कोणीतरी.
Oxidative stress.
This question is simple
Deficient
actually.
आहे म्हणजे NADPH बनेल का? NADPH नाही बनला. वन ऑफ द इम्पॉर्टंट फंक्शन काय आहे? ग्लूटाथायन कम होगा तो क्या होगा? What is one of the as an antioxidant one of the function of indirectly NADPH का क्या है? RBC सेल मेम्ब्रेन इंटीग्रिटी, राइट? तो RBC मेम्ब्रेन इंटीग्रिटी कम हो गई तो लाइफ स्पैन RBC का कम होगा. दैट्स वाई मलेरियल पैरासाइट का लाइफ स्पैन जो है वो पूरा नहीं होगा. इट रिक्वायर्स 120 days ऑफ लाइफ स्पैन, इजंट इट? ट्राई टू कोरिलेट एन आंसर ना. इतना अच्छा उन्होंने क्वेश्चन डाले हैं. प्लीज आंसर इवन इफ इट इज रॉन्ग. प्लीज आंसर.
Anyways, that's it. Any questions?
Any questions?
What is the effect of NADH and NADP plus ratio by the G6PD deficiency? And where does it occur?
NADPH and NADP ratio. NADH ratio and NADPH ratio.
NADH to
NADH to NAD plus. It's reduced from NAD.
Okay. You mean NADPH plus NADP plus?
Yes.
you are
It is-
asking NADH to NAD plus or NADPH plus NADP plus.
NADPH.
So to answer your question, it is generally estimated for the activity of enzyme. Means if NADPH is to NADP ratio is high, this shows the enzyme activity is normal. If this ratio is less, it shows the enzyme is deficient in the cell.
What is its effect in the hepatocytes?
In
What do you mean by that?
If there is the improper ratio, then what happens in the hepatocytes?
So as glucose-6-phosphate dehydrogenase is required in all cells of human body. Okay. So just like that, hepatocytes also require that, and as hepatocytes also will have that glucose-6-phosphate, it will be utilized for the conversion of glucose-6-phosphate to the glucose-6-delta-phosphonolactone. Phosphoglucono-delta-lactone, and NADPH production will be done. So the less, similarly, the less ratio will indicate the inactivity or the less activity of the enzyme.
ultimately, if G6PD is deficient, what products formed by HM patient are not produced, and what will be the effect of it? That is the answer, I do think.
Yes, sir. That's one part of answer. And along with the
सर HMP-मध्ये NADPH आणि पेट्रोल शुगर फॉर्म होतात. जर NADPH ॲनाबॉलिक रिॲक्शनसाठी लागतं, जसं की फॅटी ॲसिड फॉर्मेशन होते. जर NADPHच नसेल तर बाकीचे जे ॲनाबॉलिक रिॲक्शन आहेत त्या स्टॉप होतील.
Glycolysis will stop, basically.
नाही.
There won't be any NADPH for fatty acid synthesis. अजून काय expected आहे?
नाही सर ते क्वेश्चन अजून एक क्वेश्चन आहे व्हॉट इज द इफेक्ट ऑन युरिन क्लिअरन्स बाय दिस डिसीज
So due to the involvement of